today we have a very exciting expedition with someone who has visited Antarctica, who has done science in Antarctica. So we talk of people visiting Chandigarh and Pune, and few people we will meet who have visited Antarctica, right, and actually worked there.
Thank you to TTS India for inviting me in this amazing session. I too much like to interact with kids, because when we interact with kids, we get to know we are doing science, but at the end of the day, if the science is not getting communicated to the community, and that too to the young minds, the science for me doesn't have a sense, because ultimately the things that we are doing are not getting known to the community. When I was as a kid, I used to think that there is no life in Antarctica. There is no life in Arctic. The only life I knew was penguins, because that's the thing comes to our mind. I was really thinking someone will write penguin, but I didn't see there. But surprisingly, I was thinking that if I keep myself back maybe 16 more years in this age group, or 20 more years, frankly, I didn't had that capacity to write word like adaptive, survival, robust. So I was seeing and I was thinking that the kids have so much interest on learning new things. This really made me surprised. Okay, I'm Aparna Banerjee, and I'm working as assistant professor in one university in Chile. To give a little brief introduction, probably you all know Chile for football or for Atacama Desert. Chile is also the longest country in the world. So basically from South America, from tip north to tip south, it's entirely Chile. It have much similarity with India. In India, the north is extremely cold and the south is extremely hot. Let us make it just opposite. In Chile, the north is extremely hot and the south is the most nearest to Antarctica. The city Port William is the southmost city in the world that is the nearest to Antarctica. So it's freezing cold. And in Chile, we get a lot of environment that is similar to space. I will come next on these different models that we are working with. But when we think that how do microbes in Antarctica tell us about life in space, definitely Antarctica is not the only one. In our world, in billions of years, there have been a lot of changes. And most importantly, in last 5,000 years, as the human civilization started happening slowly, we have changed a lot. So the world was not this. The world was boiling, the world was hot, then started slowly coming down. And this process make a lot of changes in the world where the life came. So the things that we are seeing now in the world was not the same a billion of years ago. But till now, surprisingly, in some part of the world is so pristine that basically mimics the same that it was thousands of years ago or billions of years ago, or now in the space. One example is hot spring. In hot spring, it's bubbling hot water with a lot of metals, pH is acidic, a lot of anaerobic bacteria are growing over there. They don't need oxygen to grow. So this kind of life we cannot think, because we as human or the animals or the plants cannot survive over there, but the small microscopic organisms have that capacity to survive. So today we will talk mostly about Antarctica, heat in cold. So we are working on a system. When we think about Antarctica, we feel like freezing cold, but we are studying heat in cold. So this photo, I will show more this kind of photos. But it's a fumarole, if you can see, just beside Southern Ocean. So when the tide is high, the fumarole get covered totally. And then when the tide goes down, the fumarole gets up and you can see the water is boiling, 101 degree. So how amazingly the microbes are getting adapted in hours, because for some hours the tide comes and go, but the microbes are living over there. So life is surviving, and again a range of extremity, not only heat, not only cold, salinity, metals, temperature, cold and hot, pH, and all this extremity. Then another example of this kind of hot springs that we are studying in Antarctica, you can see the color. So the color you can see is brownish, dark brownish yellow color of the hot spring. That is also very near to the Southern Ocean. Interestingly, these fumaroles in Antarctica are intertidal. So when the tide goes down, we can see them. And when the tide is high, we cannot see them. So the life is surviving under heavy or trace metals because they are volcanic in origin. And thirdly, entirely this continent Antarctica is the whitest country and the country that have most windiest country in the world. So it's dry, it's windy, and it's white. What is dry for you? Probably you must be thinking how it can be dry because there is a lot of ice, but these ice are not available to take as life form to a lot of animals. So a lot of ice are permafrost and the living being cannot use them. So it is dry, it is windy, and it is white, and the life is surviving, as I told, in all these extremity, and along with that, ultraviolet, solar radiation, because there are ozone holes in the southern part of the world, so they are also surviving these factors. So imagine we, at lot of times, we as human think, I feel tired, I don't want to go out. It's so hot, I don't want to go out. It's so cold, preferably I will be now sitting in the home, taking a popcorn and seeing a movie. And these amazing microbes are surviving all those things without complaints and being happy together.
So that sounds fascinating, Aparna. So it is, the extreme is so extreme in Antarctica. You have geothermal springs, you have these intermittent tides and salinity, UV, permafrost. So already we've started having some questions, but maybe we will learn this shortly. Mohak says, how do microbes survive extreme climates and temperature? Avishai says, can only certain types of microbes survive? And Sonali asks, do you think microbial life on Earth from another planet was introduced from another planet and Earth had the proper conditions? Okay, we will take them as we go along, but I'm just trying to tell you that the chat window is really active.
The first question probably I will be sharing in a couple of minutes, how they survive.
Okay.
So when we think about astrobiology, the word initially we used to use was exobiology, life outside our planet. Now the word that we are using is astrobiology. So the life in our universe, how they are. When we think life in our world, in our planet Earth, there is life we can see. We open our eyes, we can see them. But what we don't see in our eyes, like microbes, often we tell, okay, we cannot believe it because we don't see in our naked eyes. But there is microorganism we cannot see in our naked eyes, but they are alive. So the questions arise like that. If there is biological signature, biosignature in planet Earth, can there be some biological signature outside of this world? So there's the search started. Life beyond our Earth, if they exist or not. There are a lot of Earth analogs that have been worked. Kepler is one of the example few years back, but they have they are much distantly located from the planet Earth, and when we see them captured by NASA images, they look somewhat similar. So the idea is the nearest planet that we can see is Mars. So a lot of time there is rover Curiosity, a lot of countries that have sent samples, that have sent robots over there, collected sample, sent a lot of images, and we have started analyzing or comparing those images. So how these images in Mars, or how the mineralogical composition in the soil of Mars, is it similar to planet Earth? If the water went somewhere in the planet Mars and there are some signatures of water existence a long time back in Mars, do they look similar to planet Earth? So this kind of questions arise. How they originated, if they originated, how they undergoes in this big period of evolution, how they got distributed in those planets, and what are their future, what will happen to them. So definitely one big question is, what are the factors of habitability? What are the boundaries to live? For us, boundaries to live is a temperature that is livable. There are people who live in Russia, in northern part of the world, in Norway, in Denmark, in Russia, and live in temperature near -20, -30. In Antarctica, there is no human colony. There have been a convention that make Antarctica totally dedicated to science. But in northern part of the world, the Arctic world, human is living. But they have made it. They have adapted it to live. So now the thing is, if we don't make it adapted, what are the possible analog? If there is no habitability, how to precise those analogs? Like, for example, if the temperature is less, how much less it is? If the salinity is extreme, till what the life is living, and after what life is not living and only microorganisms are living. And are they present in our planet? This kind of analogs, extreme salt, extreme pH, extreme temperature, extreme pressure, where life doesn't survive, they are present in our planet, because every day in this time of climate change, global warming, we are losing those pristine ecosystems, because these pristine ecosystems are keeping those missing link and those footprints that have been conserved in our planet Earth for billions of years. But in this time of climate change, it's very easy to lose them. And also the continuous urbanization is hampering them. Every day we are encroaching more and more lands for making them adapted to human living. And the third point is simulation. If we already know the analogs, how to simulate those factors that to find the space biosignature. So a lot of time we know that the microorganisms are living over there, we can see the color, but to simulate them in a way in laboratory that we can culture them, the microorganisms. And finally, if we have them, what are those biosignatures? There are a lot of there are till date there are more than 50 biosignatures available in this time in our planet Earth that are pristine, that are not that much contaminated with human. One of those is Atacama Desert, Chile. There have been a beautiful publication in 2000 in Nature that demonstrated how Atacama Desert in Chile is a Martian analog. So the astronauts of NASA came over Atacama Desert in Chile, and the astronauts were making a pseudo living in Atacama Desert as if it is similar to Mars. Deception Island, Antarctica, one that we are working now, Rio Tinto in Spain, Atacama Desert in Chile, just to let you know, it's also the driest point in our planet Earth, the most driest point in planet Earth. Deception Island in Antarctica, we will talk more about this. Rio Tinto in Spain is totally red. So if you go and see, there are a lot of deposition of iron, calcium, salt, and these salts make the river red. Saltenskov in Denmark, Mojave Desert in United States. So these are only few examples. You can search about this, and this will grow so much interest in knowing how life is surviving in this kind of environment.
So Aparna, before we proceed, one question Bhavik should clarify is, what are biosignatures? When we say these 5 places are space biosignatures, what are biosignatures?
Where there are biological, what is signature? That you come to know that this is living, isn't it? There is some proof. So there are these signatures of biological life existing. So it's not that they are not explored, they are explored, and it has been seen that other than plant life and animal life, the life that has not been seen, extremophilic microorganisms are surviving over there. So why we are calling them biosignature, like space, because in space the extremity can be radiation, the extremity can be very high temperature, the extremity can be wind, the extremity can be pH, and or a lot of heavy and trace metals. And this kind of environment are present in these localities that I mentioned, and more than those. And there are microorganisms surviving in those kind of localities.
Okay, so like the conditions mimic Mars or mimic space, but there is life in these places.
There is life.
So it helps us to study the possibility and potential of life in space. Okay, great.
Because we cannot go to space. There are a lot. Till now, there have been no human that went to Mars. Till now, there have been no work. So the work that has been done till now is simulating the possible life that rover Curiosity or other robots have sent the samples, the images. And there have been a lot of publications about the hot springs in Chile, about Deception Island in Antarctica, a hot mountain. So how they look similar. So it's just comparing the environment in planet Mars or some other extraterrestrial environment, and how these both look similar together. So this is the way we are seeing an analog, and we are simulating those environment to see that if life is existing over here, possibly life is existing in planet Mars too.
As well. Okay. All right. Thank you. We'll move on.
Okay. So when we are talking about all these Actually, the part is that the upper part I cannot see for zoom, and I really don't know how to make them small.
Okay, you can just move it. Usually that bar can just be moved.
No, not that bar, in the black thing over.
Oh, if you want, I can also share it quickly.
It's going now back, and no, it's going.
No problem. No problem. If you shall I share?
It will be better.
Okay,
Please.
I will share. Yeah, I think you were here, no? Aparna?
I was here, precising the analogs. Yes.
Go slideshow mode. Yeah. Yes, perfect. Go ahead. And you tell me when next I'll keep moving.
Yes, okay.
Yeah.
It's showing loading till now.
Okay, it's showing for me. Shreya, is it showing yet? Oh, why is it paused? Let's try again. 1 minute, huh? Just a minute.
Mm-hmm.
Yes, I think it should work now. Yes, it's showing?
Now showing, yes.
Okay, great. Yes?
When it's going to the full slide,
Yeah,
then
it's
only
not showing, no?
loading,
Exactly.
yes.
Okay, 1 minute. I think, yeah. No, there's some issue then. Aparna, what we can do is for time being, we just show it in the partial slide mode.
No,
Let me just, I can.
Sorry, everyone, just give us a minute.
I'll be going back there. I'm just
okay.
starting from there, precising the analogs.
Okay.
Yes,
can you see
Yes.
my screen?
Yes.
Yes.
Okay.
Yes, great. Thank you, Sneha. Thank you. Yeah.
Okay.
Go ahead, Aparna.
Okay. Thank you so much. So we were in the part, we were thinking of precising the analogs. So we were talking about different possible analogs, and precising the analogs, when we think about few main factors that comes to our mind. The first factor, temperature. So Mars was once a warm and wet planet, and today it has changed into dry and cold. And similarly, if we see that Atacama Desert is a dry and a cold space. So if we are thinking to see an environment that can be somewhere similar to Mars, we can see Atacama Desert. That there can be one factor, radiation. In outside of our planet Earth, there is solar radiation, there is galactic cosmic radiation in Mars. In our planet Earth, there are so much like Mars, but there is ultraviolet ray, solar radiation present. And in the southern part of the world, because of the ozone hole in stratosphere, there are stratospheric microorganism has already been discovered. So this is not something that we are comparing. Actually, there have been balloons sent to stratosphere, and the balloons bring back air from stratosphere. And this work has been done few group of Indians in ISRO. So there are amazing microbiologists even in India who has worked on a lot of microorganisms on stratosphere. And those microorganisms have been observed in different other conditions in planet Earth too. So it's not that stratosphere have something special. The microorganism that is surviving in stratosphere, the microorganisms are available in our planet Earth too. About Mars that we were talking till now, no, directly with the sample of Mars, Martian soil has not been worked. But till now, the work that has been done, we know the mineralogical composition that has been done. So we know what are the possibilities in Mars. And we try to create the similar kind of possibilities in planet Earth, just to understand what are the boundaries of life, and how outside these boundaries of life is surviving in Earth. So if it is surviving, it's a question mark. Are they really surviving in extraterrestrial environment too? So when we were talking about radiation in Earth, in the southern part of the world, there is, because of ozone hole, there is ultraviolet radiation too. Another point, desiccation. So once Mars, I'm giving the example of Mars because we lot have a curiosity about Mars. If I give one name, desiccation is dry, yes. So once Mars used to run red with water, and now they dried up totally 3 billion years ago. There are those signatures that as if the water was going through there, and we know all that rover Curiosity have sent those images, NASA have published those images. So it's like it was a river, And when the river dried up, those kind of movement of river that have been seen in Mars. So in this kind of environment, when we see in Atacama Desert, in Mojave Desert in United States, there is this kind of dried up river that can give us an idea about driest point in planet Earth that doesn't need water and microorganisms are surviving. And the fourth point, definitely, a lot of other planets, almost all the planets have been formed because of some volcanic activities, eruptions. A few planets breaks up into two more planets. So during all these volcanic activities, a lot of metals and metal ions, metal salts are there in extraterrestrial environment. So for example, in planet Mars, it is already detected. There is a lot of iron. We see planet Mars red, of course, silicon, magnesium, aluminium, calcium, boron, and all these kind of metals. A lot of metals are this kind of metals are present in very high level in a lot of this kind of analogous environment. For example, we are working in one of the ecosystem in Chile where we see that, I will show the figure images later also, it's totally red, the hot spring. So there is a lot of deposition of iron in this kind of places. There is a lot of silicate present in this kind of places. So if life is surviving in this environment, again, there is a possibility that life is surviving outside our planet too.
So Aparna, this leads us to a nice point asked by Meher. So do you think, right up front, life can be possible on Mars and we are only waiting to find it? Because of so many analogs that we have managed to map?
Actually, in science, what I believe that there is, we cannot see, yes, we cannot see neither no, but we having a lot of friends that are working directly with astrobiology. I'm more into like how they survive, how microorganisms are surviving, adaptations of microorganisms in this kind of environment. But I know a lot of friends of mine who are working on microbial taxonomy, who are trying to find new microorganisms in this kind of environment. And they really believe what I heard from them, that this kind of environment give us an idea. Actually, I had a very nice experience in Antarctica this time. We had a Colombian group and they're very famous in Colombia. I have seen them giving a lot of interviews and this in Colombia. They're young couple and they're astrobiologists, and they had, out of their focus in the hot springs, they had one more focus. They were taking interview of the Argentina Navy and the Spanish Navy to ask them what is their experience to live in Antarctica for 3 months, isolated in a base, extreme environment, and almost without communication or without internet for a really long time. So they were telling that this is the word, they are having a pseudo astronomical experience somewhere isolated in our own planet Earth. So they are working on this, they are doing their PhD on this, and I find it so interesting that I was thinking, okay, I'm really a pseudo astronomical person. And so it's like there are possibilities.
So it is like a pseudo space environment, basically. Space environment. So who knows? Like Inesh asked the same question. Can we conclude that life might be possible? Might. There are lots of analogs. Okay, Aparna, you go ahead. Yes.
The next, please. Okay, so the extremophilic microorganisms, they are breaking the limits of life every time. There are a lot of extremophilic microorganisms in different kind of species in planet Earth. Particularly the one that I am looking into, I have more interest on temperature, the variance of temperature. So we have worked into one hot spring in India, Panipala, that is in a volcanic zone, Chotanagpur volcanic zone, and this volcanic zone is complex. Pressure has been worked a lot. This hot spring was not worked earlier. So it came, the geothermal expansion came because of mining activity. There is thermal manifestation. And interestingly, it was having 433 ppb of arsenic. So till date, the highest that has been reported from India is 130 from a Himalayan hot spring, and it's called arsenic reach. And we were surprised that this hot spring had almost 3 times higher arsenic in the hot spring. So this is out of other different metals, the arsenic came into our mind for this, that okay, how the bacteria is surviving in this high arsenic contaminated environment. Out of different other works, we got a bacteria Brevundimonas, and this was surviving the exposure of arsenite and arsenate really very high. Out of arsenite and arsenate, there is arsenic 3 and arsenic 5, the valency I am talking about to you. And there are a lot of examples where microorganism can tolerate arsenate, but arsenite is a really toxic form. And this bacteria was tolerating arsenite till 90 millimolar, which is really high and have not been reported till date. So this is the reason I always think that the extreme environment have immense possibilities. The more we can talk about conservation of those environment, conservation of the microorganisms in their own environment. We talk a lot about conservation, but when we talk about conservation, we talk about plants and animals. Rarely we talk about microbes, and they are really surviving in those. And the science never ends. So every time we work more, we get to know something more. If we are talking about this Campanario in Chile, that we another analog that we are working now at this moment, this is in Andes mountain, and in Andes mountain there is there are a lot of volcanoes, active volcanoes. Chile have more than 400 volcanoes. Some are active, some are not. This Campanario is a great volcano that was active 3 billion years ago, and it's again a thermal manifestation. And if, as you can see, the hot spring is totally red and it's always bubbling. The temperature is around 64 degrees. It maintains the temperature. How surprising it is that for billions of years, the water is coming out. The color is red, and because of very high temperature, the animals or the plants don't grow nearly, but these biofilms are beautifully structured over there. In India, often we have this kind of thing. If water comes out, we make it a tourist place. In Chile, no, we try to keep it. In a lot of other countries, we keep them a model environment, educative environment for general people. So this kind of places, the microorganisms we have seen, and we have seen how the microorganisms are surviving, what the biological compounds they are producing, that they are surviving in this kind of environment. We have seen that in this hot spring, strontium is present, and strontium is a very toxic trace metal. So it's just an example that there are a lot of things that we don't know life can survive into, but they are surviving in this kind of environment. And the third that we are talking now is Deception Island, Antarctica. We recently went. We have been there for around a month, a little more than a month, and it's an active volcano. The Deception Island, I'm coming more into Deception Island then. So, okay, the microorganisms are surviving in temperature, in pH, in water scarcity, in salinity, in pressure, in radiation. All this kind of environment are present in our planet Earth, where plants and animals doesn't grow, but the microorganisms are breaking the limits of life and are omnipresent throughout those ecosystems.
So Aparna, that's a great point that there's so many extreme conditions. Avisha has a great question. Can you take a microbe from, say, Atacama Desert or this Campanario in Chile, take it to Antarctica? Will they grow?
There is a factor in this. There are about shifting biological beings from one country to another country. Earlier, this has been done a lot. If we see now in India, there is Parthenium. Parthenium is an introduced species. So this introduced organisms, yes, they create a lot of problem. So after that, in last 50 years, there have been a lot of laws that you cannot introduce an exotic species in another environment. So yes, we can definitely create or mimic an environment to see if they're growing or not. The question that was you were talking Campanario have a temperature 64 degrees. So let me think that it's growing. It has the possibility to grow in a fumarole in Antarctica if the Antarctica fumarole have a temperature 64 degrees. So just to give an example, in the Campanario, we got Bacillus ionaceae bacteria that was earlier reported in very depth of Pacific Ocean. So a bacteria that is growing in deep of Pacific Ocean, that is cold and pressure, that is growing in high temperature, metals, salinity, ultraviolet radiation in a hot spring.
Okay, so there are ethical implications. You can't just pick it up in your bag and put it somewhere else. Okay, sounds good. We can move on. Yes.
Can we go to the next slide? Okay. So are these extremophiles are adapted to best fit? Like how they are, I saw a lot of questions these, how they are surviving. So this is like how they survive in this kind of extremity. If I put a hot water in my skin, I will have a 3rd-degree burn. I had 1 year back 3rd-degree burn. So how the extremophiles are surviving in this kind of burns, in this kind of salinity, in this kind of extreme temperature, extreme pressure, extreme metals, so if we give an example of us, our mom probably say a lot of time to us, okay, like तुमने कभी पढ़ा नहीं ना ऐसा सिचुएशन में, पढ़ने दो ऐसे सिचुएशन में खुद भी कुछ सीख जाओगे. So it's like a lot of time our parents say when we complain about life, they say us or not, that when we will fall in a difficult situation, life will make you learn by your own. The microorganisms are like that. When the environment is smooth, the microorganisms, if fitness trait and environment, the environment is smooth, they don't grow to be fit. But when the environment have a lot of extreme, they grow to fitness. So because of this extreme, they make an evolution in a way that they're adapted to best fit. Now there are a lot of idea that when we increase stress, they are becoming fit. But what are the things that they are doing? Okay, so they have membrane changed. They have adapted the composition of membrane in a way, the cell membrane, that they can survive this kind of extreme environment. They produce a lot of stress enzyme. They produce a lot of pigments. They produce polysaccharides or biofilm. We talk a lot about biofilm. Dr. Kaushik we have here, who is an expert on biofilm. I work a little on this. My focus is more on polysaccharides. But these biofilms, under environmental pressure, the microorganism, they produce this kind of biofilm that permit the microorganism to adhere to some substrate, and a substrate is really necessary. Imagine if just to give an example of our own bacteria in our body, Escherichia coli, it's a very helpful bacteria, we know. But when Escherichia coli infect our urinary tract, it needs to adhere to the tract to grow, and that's pathogenic. So to be pathogenic, they need this adhesion. Imagine if they cannot adhere to with their fimbriae or pili to the substrate, due to the flush, they will get out. So the adhesion is really important, a very important factor in an extreme environment, that the microorganism get a house. Imagine you are outside in the environment, you don't have a house, you don't have a roof over your head, how can you grow? The same for the microorganism. You have a substrate and you are making your own home. So imagine you have your land, then you put one bricks, and then you make an entire home, and then you invite your friends also that come, I have made a house, you come and live with me, because outside is very hard for you. And the microorganisms are like that. Often they make multi-species, multi-genus biofilm, and inside one's place, they all live together. We human have a lot of complaints about life, but the microorganisms are really adapted in a way that when they are adhered to a substrate, they divide and they make more biofilm, and when they are a lot together, they get detached and they go to another place to grow.
Right. Okay, so Aparna, you're saying that in these extreme environments like Antarctica, microbes do form biofilms. They live in communities. Yes. Okay. And so that's also an adaptation to the extreme condition.
Extreme environment, yes.
Sounds good. Okay.
Yes. So this term biofilm is a complex term. Just to give a little idea on this, often we confuse biofilm with polysaccharides. Biofilm is actually the house, and polysaccharides are the bricks of that house.
Sure.
So the biofilm is having that matrix and microbial cells with some polysaccharide, and inside this matrix there are polysaccharide, lipid, protein, a lot of things. And again, these polysaccharides are made up of some monosaccharides, and with monosaccharides some groups, additional groups like acetate, pyruvate. So this is just an idea how the bacteria make their own house.
Okay, Aparna, we'll keep a check on time. Okay. We should move on a little bit. Huh?
Okay. Yeah. So now to come to the story of Antarctic Deception Island, I took this image in 22nd of December 2022 when I was in Antarctica. It was taken by my iPhone. I just saw that the screen came in iPhone and I just took a screenshot of that. And I was just thinking, okay, I'm really in the southern part of the world and this is how they look. So it was looking that I am really in the South Tip and the world was there. I was feeling okay where I am. So I just wanted to share this, like this internet thing is really crazy. It often makes us surprised how they give us so many surprises a lot of time. So this part that we can see, Antarctica have one tip. We were in that tip. This is called Antarctic Peninsula. Antarctic Peninsula have a lot of islands. Out of those islands, one is Reception Island. You can see it have a horseshoe shape, isn't it? It was earlier not a horseshoe shape. A number of years ago, there was a lot of volcanic activity in Deception. Deception was a mountain. Had a volcanic activity, make an eruption, become like this, the water entered and now it's like this. So there is water now covered. Basically, it's just a crater. So a crater filled up with water, water of Southern Ocean, is now an island. Earlier was a mountain. So this is how the island look like, the down. And what are the factors that are there in Deception Island? In Antarctica, extreme cold temperature. The coastal wind is very high, sometimes even till 300 kilometers per hour. During the time that we couldn't work 4 days and we had 130 kilometers per hour of wind. It was really difficult even to stand. So we were like moving like this. And in this kind of environment, we cannot go outside of the base. So we have to be inside of the base. So when we go to Antarctica, you don't know. You don't know which date you are going and which date you are coming back. It's always a possibility. They always give a 7 days window and a 7 days window. And when the weather will be good, you will go on that day. When the weather will be good, you will be back on that day. So I had my date 5 days delayed and I had a date 2 days more to come back. So it's never sure when you can really travel to Antarctica because of the wind. There are blizzards. And these blizzards are really risky. And it's more risky because Deception Island is volcanic origin and have pyroclast. So if it's blizzard, you are getting those snow and suddenly you are getting those small sand particles also on the face. So it's more risky and you cannot go. And there are glaciers. So never you know where the snow is slow over there and you just put your leg and it's pyroclast and it's risky. You cannot go out often without the permission of the Navy people. It's an active volcano and there are a lot of fumaroles. There are animals that live in this kind of area. For example, the seals. The seals come in a particular time period only in the summer, Antarctic summer in February. So February is a comfortable environment for them when they take the shore, because you can see that this have a lot of Antarctic Peninsula islands have mountain, but because Deception Island have beach, the seal prefers to come and take some sun during the time. There are also penguins, there are a lot of small animals, there are butterflies, there are mosquitoes. So this kind of small animals are there. But let me go to the next slide and I can let you know what are the environment that we are.
Sure.
Okay. So when we went to Deception Island, our interest was not the snow. Our interest was how near the sea these hot springs or these fumaroles are active. So you can see that near the sea, it's totally near the sea, and the water you can see is vapor coming out and it's very high. In one of them you can also see little yellow color, and these fumaroles are forming the biofilm of bacteria. So we are now analyzing those, what can be the microorganisms that are surviving, but we just went a month back. So we have discovered the bacteria are there, but we yet don't know who are they. So the names we are on that process to know. So we were trying to see what are the temperature stretches from what area to what area the temperature is high, what are the temperature range in each of the point. And there are vents for hot gases. The gases are sometimes sulfur, sometimes carbon dioxide. So when there is no sulfuric smell, there are bubbles, the water bubbles come out and you don't get a sulfate smell, it means there is carbon dioxide present. So can we go to the next slide? And when we see this kind of environment, we try to take a closer look. So from far, we see hot vapor is coming. And as you see, it was not, we were living in one part of the Deception Island. We cannot walk because of the pyroclast. We have to have a Viking equipment. We have to wear almost 20 kg, big, huge Viking dress. and literally looking like astronaut or polar bears, red polar bears we move. So it's really difficult to even see it. It's difficult to work because if our body weight is 60, 50, include 20 more into that, then include the water that we are taking in our bag, the foods and everything. So if your weight is 50, you are taking almost half of the weight with you because of the dress. And you need three, other than the, you need three layer of clothes. First layer, protective layer, the second layer, and the third layer is the protective jacket because of the wind and the rain. There was a day that we were going to the end point of Deception Island. We were in this point and the horseshoe another end. The circular water movement is very dangerous. And we worked for 6 long hours because the stretch of the fumarole was really big. While coming back, a lot of time our bike was trying to go to our base and was not able to come because of the tidal movement, and I almost lose my sense in the hand. So in my life, I really don't remember when I last cried, but that was the time I really felt fear for my life and I was crying. The moment passed by and I took a lesson from that we cannot wear a gloves that feels like warm. We have to wear a gloves that don't feel much warm, more like thin polystyrene material, but if the water enter, water will maintain a temperature. So it's like not often you can think about your own commodity. You have to also think what can be good if you don't have food, if you don't go back to the base for a long time, if your material can sustain the extremity or not. So this is the kind of environment that we were looking into. One image you can see green and yellow beautifully. So the biofilm was totally separated from one another. In another, you can see some yellow color, red color. So we were taking biofilms from that part of environment. So the microbial community, we wanted to see what are the feedbacks they are doing with the environment, because the environment have ultraviolet C radiation, salinity, extreme pH, extreme temperature, high concentration of heavy metals and trace metals, and in all this extremity, how the microbial community are interacting with one another. If they are interacting, if they are producing polysaccharides, they are forming a home, they are making these building blocks, and how to use them. So we are also thinking into circular bioeconomy, white biotechnology, that can we use those microbial bioactive compounds for our own human use? So that's our final goal. The next slide, please.
Sounds good. Okay. Next slide, Sneha. Yes.
So there is life. So I'm just showing one image. This was from that water bubble that you see just near the ocean. So we took and we make some confocal microscopy, and this red point that you see is we make a confocal microscopy staining with a particular stain that can bind to polysaccharide and that can bind to bacteria. So the bacteria, you can see a lot of bacteria in that 101 degree bubbling water. And there is polysaccharide also, you can see. It is actually a video. I didn't have the possibility to share the video, but when the video was moving, the confocal microscopy show you the images in different fields. So there was a long string of polysaccharide there. So microorganisms, even in this kind of environment, is producing polysaccharide. So the temperature was sub-zero in the vicinity because the water is coming and going down in Southern Ocean, but boiling water in the fumarole. We just make a little hole and the boiling water 101 degrees centigrade started coming out. The pH varies from slightly acidic to slightly basic in different fumaroles. We worked with 8 fumaroles, all the fumaroles in Deception Island. There has been work separately in 1 or 2, but this time we had almost a month, so we took samples from all. And there was high electrical conductivity in the water that indicates presence of metal ions, different salts, and we can see bacteria were there. So we have already isolated few bacteria. One of the bacteria is dark black, and when we open the culture, it gives a sulfuric smell. So there are a lot of bacteria that are really interesting we are working into, and they're sustaining. Microbes are there. They're sustaining this extreme temperature, pH, metals, UV light, salinity, and all.
Okay. Quickly, Aparna, Avishay asked, what is the red dot? Since you're short of time, What is the red dot?
That red dot is a string of polysaccharide. We stain it with a stain called cyproruby that bind only with the polysaccharides.
So it's the polysaccharide part of the biofilm.
The part of
the matrix, what we call. Okay. All right. Lots of questions, but we'll move to the next slide now.
Okay.
Yes.
I think I do not have next slide.
Okay.
It's just an acknowledgement, and I really wanted to show these images of our group because we live a long time together without our family for almost a month, and they all become our family. When we are in our country, we think about, look, a person from that country. But for all this time, we had people from Colombia, from Chile, from Spain, from Portugal, I from India, I'm working in Chile, and we never fell for a time. So the world is unique like the microorganisms. They all live together, and we all were surviving in a country, in a continent, Antarctica, that is not owned by any country.
True, exactly.
And it was an amazing experience.
I was just going to come to that it's so appropriate for Antarctica because it is still not owned by anybody.